Hi everyone, it's Ms. Lopez. Welcome to your notes on Chapter 6.2, Part 2, the Krebs cycle. In this section, we are looking at carbohydrate catabolism in the presence of oxygen. We have already started with the first biochemical pathway called glycolysis, and at the end, it produced pyruvate. In the second video, we took that pyruvate and converted it into acetyl-CoA. That process occurs in the mitochondria. We're now going to take that acetyl-CoA and see what happens to that molecule. But first, a quick overview of where we are going. We've already seen how the role of glucose in the breakdown process called glycolysis starts this reaction. We've even seen some of the carbon dioxide that comes out of the reactions when pyruvate is converted into acetyl-CoA. We're going to see how the rest of the molecules in this reaction fit in, including the oxygen and, most importantly, the water. This last biochemical pathway is called the Krebs cycle. So we started with glycolysis in the cytoplasm of the cell, breaking down glucose into pyruvate. We took that pyruvate and sent it to the mitochondria where it is being oxidized. Now, at the end of the pyruvate oxidation, that ending molecule called acetyl-CoA is going to enter into what is known as the citric acid cycle. This is also sometimes referred to as the Krebs cycle and also referred to as the tricarboxylic acid cycle or the TCA cycle. Please be familiar with these three terms as we don't know which one College Board is going to ask. But either one means the exact same cycle, and this is going to occur in the matrix of the mitochondria. A quick reminder of what we've produced so far. In glycolysis, we produced 4 ATP through substrate level phosphorylation. We produced 2 NADHs when we reduced NAD plus down. We've also produced or reduced NAD plus during the oxidation of pyruvate, and so now we have 2 additional NADHs. And we're going to see what the Krebs cycle produces to add to this slide. So let's begin. Acetyl-CoA, which was produced at the end of the oxidation of pyruvate, is going to undergo a series of biochemical reactions. So this is a biochemical pathway, just as we have seen before. There are several steps involved, as you can see illustrated on this picture, and each of these steps is catalyzed by an enzyme. Now, you don't need to know the names of these enzymes, nor do you need to know the names of these molecules, with the exception of possibly 2. This right here, citrate, is the first molecule that is produced when that acetyl-CoA joins with the end molecule of the Krebs cycle. So it's called a cycle because what you start with needs to be regenerated as the last molecule so that you can start the next turn of the Krebs cycle. So with acetyl-CoA joining oxaloacetate, it forms citrate. Now take a look at our carbons. We've got 2 carbons on acetyl-CoA. Oxaloacetate has 4 carbons, and so what you see in citrate is a 6-carbon molecule. Now this citrate is going to be converted into another product that is catalyzed by an enzyme. And then this product becomes the substrate for the next enzyme. And so this is just a biochemical pathway. But what you'll notice is that sometimes we change from a 6-carbon molecule to now a 5-carbon molecule. And the way that we do that is by releasing carbon dioxide in the process. This reaction, or this enzyme, sometimes needs the help of our friend NAD+, which is reduced down to NADH. You can see that this happens 1, 2, 3 times during the Krebs cycle. We're also losing carbon dioxide, and in this process, we are producing an equivalent of ATP known as GTP. This process is still going to be made as a substrate here, is being converted into a product here. So this is still substrate level phosphorylation. And you also hopefully had noticed that a molecule of an FAD is going to be reduced down to FADH2. We'll talk about these molecules and what happens to them in our last section of aerobic respiration. As the molecules, as the substrates become or get acted upon by these enzymes and become products, and then that product gets acted upon by the next enzyme, this process will continue until you get your final molecule, which is called oxaloacetate. That 4-carbon oxaloacetate will then join with a new molecule of acetyl-CoA to keep this cycle going and continuing. So during the Krebs cycle, all of this is occurring in the matrix of the mitochondria. Acetyl-CoA enters, and the Krebs cycle remakes the starting molecule, known as oxaloacetate. It produces ATP by substrate level phosphorylation, although in your video they show it as GTP. The GTP can phosphorylate ADP to produce ATP. We're also going to produce NADH and FADH2. Now, in that previous slide, we saw that there were 3 molecules of NADH produced and 1 molecule of FADH2 that was produced. But what you saw on that slide was for 1 molecule of acetyl-CoA. In other words, for every molecule of acetyl-CoA, we produce 3 NADHs, 1 FADH2, and 1 ATP molecule. But keep in mind that 2 acetyl molecules, acetyl-CoAs, are produced from 1 glucose. That means that 1 glucose molecule will cause a turning of the Krebs cycle twice. So from 1 glucose molecule, we are going to produce a grand total of 6 NADHs, 2 FADH2, and 2 ATP through substrate level phosphorylation. So I have updated this chart where we're keeping track of all of these molecules that are made. And we're going to see how they're important when we finish up aerobic respiration with the final section and our oxidative phosphorylation. This concludes the Krebs cycle notes for chapter 6.2. Thank you all so much for watching.